Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to the latest Novogene webinar, Non-Coding RNA Sequencing Results Explained, what you can explore with non-coding RNA data. This webinar is part of our ongoing Beginner's Guide series that includes webinars on RNA, DNA, and bioinformatics. You can find all of our previous webinars on our website, and we'll be emailing details of our upcoming webinars to you. A bit of housekeeping before we start. The webinar will run for a total of 1 hour with around 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of the screen. You can upvote a question by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. If we don't have time to answer your question, we will come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We will share the slide deck and recording of the webinar via email. You can expect this on Friday. If you're studying and you need a certificate of attendance, there'll be a link in the follow-up email that will display the certificate that you can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this, as it helps us to plan future webinar topics. I would now like to introduce your host for the webinar, our scientist content specialist, Aoife. Aoife, over to you.
Thanks, John. Morning, everybody. My name is Aoife. I am the scientific writer for Novogene, as John mentioned, and I will be your host for today. Apologies about that. So Novogene was founded in 2011 and has been growing rapidly since then. So our company mission is to advance genomics and improve life. And as you can see here, our services cover a huge range of applications with real life implications, from complex disease research and oncology to food and water safety. Our European Sequencing Centre is based in the Cambridge Science Park in the UK. Here we have our state-of-the-art lab equipped with the latest Illumina NovaSeq 6000 sequencing machines. We have always provided fast turnaround times and high quality data, but this is set to improve even further with the installation of Falcon 2, which is our new intelligent sequencing platform. And I will touch a bit more on that later. We also have our supercomputing cluster, so we can offer bioinformatics services and store your data in the UK. However, at heart, we are a global company. We can offer services across Asia and the US and, of course, Europe, with almost 2,000 employees and serving over 5,600 customers in world-class universities, biotech companies, and hospitals. We can provide sequencing for the smallest or the largest projects. We provide a full range of sequencing services, such as whole genome sequencing, whole exome sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. These can be applied to a wide range of research topics, from human disease, microbial cell factories and gut health, to agriculture and animal health. The focus of today's webinar is on our non-coding RNA sequencing service. Non-coding RNAs are defined as RNA transcripts that don't get translated into protein and include long non-coding RNA, small RNAs, and circular RNA. These molecules have been shown to play a role in processes such as the regulation of transcriptional machinery, gene expression control, splicing, and DNA replication. We can sequence each type of non-coding RNA individually or combine this with our mRNA sequencing service for an analysis of the whole transcriptome to assist you with building an RNA interaction map for your cell line, your testing conditions, or whatever species you're studying. The NGS market is crowded, but there are a lot of good reasons as to why you should trust Novogene with your project. We have had a really exciting development recently, as I've touched on earlier, in the installation of the Novogene Falcon 2, which is our new intelligent NGS delivery platform. This new technology was developed in-house entirely by Novogene and will massively increase the capacity of the Cambridge Sequencing Centre. It will also increase the quality of the data produced, as it is entirely automated, and will remove the possibilities of human error. With this new addition, we are now able to offer our fastest ever turnaround time for RNA sequencing, which is just 10 working days from sample arrival to data release. The Falcon 2 really highlights the innovation that we strive for here at Novogene in an effort to keep pushing the frontiers of genomic research. The benefits of our in-house innovation are clear in our record in the scientific community, which we are very proud of. We have been cited in over 790 research papers with a total impact factor of 6,120. We are also driving the NGS industry forward with over 37 NGS-related patents. Our technology is helping to make the NGS process more efficient and effective, all the while improving the quality of data that we deliver. The Novogene community keeps on growing. Our expert sales team speaks over 10 languages and are located across Europe, ready to assist you in your next project. If you are not ready to send us samples yet, you can still benefit from our educational outreach to learn how NGS can work for your project. We recently wrapped up the first installment of our European Roadshow, where members of our sales and product teams delivered seminars about the applications of NGS in DNA and RNA research. So keep an eye out on our social media for future seminar announcements, and hopefully we'll be visiting somewhere near you soon. Let me just quickly introduce the sales team who are based all across Europe. We have our UK and Ireland team, our DACH region team, and our Benelux team. The team has grown so much recently that it takes 2 slides to cover everybody. So here we have the Southern European team, the Nordics team, and the Eastern European team. If you would like to speak to your local representative, please do contact us via our website and we'll be able to put you in touch with them. With that, I would like to thank you for your attention, and I will hand over to the presenter for today, Carolina. Carolina holds an MSc degree in computational biology and is a product marketing manager with Novogene. Within that team, she is in charge of eukaryotic RNA and non-coding RNA sequencing products. So with that knowledge, she is the one who will guide you through the bioinformatic pipeline for non-coding RNA sequencing today. So over to you, Carolina.
Thank you, Eva, for your lovely introduction. I hope you can see my screen right now. And once again, thank you all for joining our webinar about non-coding RNA sequencing results, where you can explore with non-coding RNA data. So to start, I'm going to give you a brief introduction about what is non-coding RNA sequencing, and we're going to see the different types of non-coding RNA, and of course, the Novogene solutions that we can provide in order to study them and isolate them. Then we'll have a detailed view about a non-coding RNA sequencing bioinformatic analysis service workflow, which will include steps like quality control, mapping or alignment to the reference genome, annotation step, and analysis about quantitative, functional, and qualitative approaches. Once we have a detailed view about the non-coding RNA sequencing pipeline, I will also show you some highlights in terms of small RNA sequencing and circular RNA sequencing. And last but not least, I will finish the presentation mentioning the potential interaction between the different types of non-coding RNA and how to establish these regulatory networks. So let's start. And what is non-coding RNA sequencing? So it is actually a part of RNA sequencing technologies where it uses next generation sequencing tools to get a snapshot of your sample. This snapshot is basically going to focus on the transcriptome, and the purpose is to reveal the presence and quantity of RNA in a biological sample at a given moment. So here in this figure, you can see the different types of RNA. We have messenger RNA, which usually encodes proteins. We have non-coding RNA, which can be divided in housekeeping, and also regulatory non-coding RNA. And within these regulatory ones, we can also distinguish them by their size, and they can be considered as non-coding RNAs and also small RNAs. So these non-coding RNAs that have regulatory function, they're well known to be actively involved in gene expression by either regulating transcription or post-transcriptional modifications. Now, we're going to start by introducing the different types of non-coding RNA that have regulatory functions. The first one is the non-coding RNA, and it comprises of non-coding RNAs that exceed 200 nucleotides, and they do not have coding potential. This means that they are not going to encode proteins. And based on the genomic position, we can distinguish them in intergenic non-coding RNAs, which usually account for the largest proportion within the non-codings. And also we have intronic, sense, antisense, and bidirectional non-coding RNAs. Now, non-coding RNAs, they are well known to be involved in multiple cellular processes, and they interact with different kinds of molecules. So we can define this as a sort of non-coding RNA interactome. And they're involved in gene transcription, post-transcriptional modifications, and also in epigenetics. So non-coding RNA, they can interact with proteins, with RNA, and also with DNA, And it is a very important field where we can use NGS tools to study diverse biological functions and how they are involved in diseases such as cancer or neurological disorders. Now, if you want to study long-non-coding RNA, usually the recommended cDNA library that you need to build has to be strand-specific. So you have your total RNA, and you're going to get rid of the ribosomal RNA and fragment it in the pieces in small fragments, usually ranging between 250 to 300 base pairs, and you'll move on to the cDNA synthesis. Now, the characteristic of strand-specific libraries is that during the second strand cDNA synthesis, you're going to replace the TTTPs with the UTPs, this strand that is in red. And once you have the adapters ligated, that's when you're going to digest that second strand that has the DUTP through an enzymatic digestion. And you will end up with a library that only contains information about the first cDNA strand, which is basically the antisense strand. And now during the data analysis, you will be able to tell apart sense and antisense transcripts. Now, moving on, we have seen the long non-coding RNAs. Let's see the short ones. They are known as small RNAs. They are short molecules. They are non-coding again, and most of them are involved in gene silencing and post-transcriptional regulation of gene expression. So by studying the small RNA sequencing fragments, we are able to have a look at these short RNAs, and especially the microRNAs, and we can analyze the differential expression of microRNAs. We can predict potential novel functions for these small RNAs, and we can also see potential changes in their structure. So now talking about microRNAs, which I probably will mention them as miRNAs from now on, they are one of the most well-known types of small RNA. Usually their size ranges around 22 nucleotides and can be actually a range of 15 to 34. So they are formed by this cellular process. And when we have the mature miRNA, they can bind directly to the messenger RNA. And depending on that binding, we can have two different reactions, but in the end, it's going to cause RNA silencing. So for instance, if the mature miRNA is going to have a near perfect complementation to the messenger RNA, that will cause a messenger RNA degradation. And on the other hand, if it's going to partially complement the strand, then it will just cause a repression on the translation process. Now, moving on to the third part, we have the circular RNA, which are actually one of the most stable types of non-coding RNA. They're single-stranded, and they form a covalently closed continuous loop. Thanks to the circular structure, it makes them really, really resistant to exonuclease digestion, which is a feature that we're going to exploit when we build the library preparation. Now, most of these circular RNAs, they arise from back-splicing protein coding genes, and they have several functions. But the most well-known function is acting as miRNA sponges and also as protein decoys. Now, what can Novogene offer you in terms of isolating and sequencing the data of these different types of non-coding RNA? For instance, if you're interested in studying non-coding RNA sequencing, we recommend the ribosomal RNA removal library. And by getting rid of the ribosomal RNA in the raw data, we will actually have not only long non-coding, but also messenger RNA information. Now, the strand-specific library is provided by default, and the sequencing strategy is following the pair end 150 using Illumina NovaSeq 6000. As an optional feature, if, for instance, you're working with blood samples, let's say there are RNA extracted from human blood or maybe mouse or rat, we can also get rid of the globin messenger RNA using the globin kit. And if you're working with exosome samples, we actually have a customized library prep protocol using a different kit that allows us to take in these kind of samples and work with them even if they have a low input. Now, additionally, if you're working with cell cultures and you have RNA that needs to be sequenced, in most of the cases, it's going to be fine. But if you suspect a potential contamination from bacteria that is growing in the culture and cannot be detected by the naked eye, we can provide a dual ribosomal RNA depletion, because by default, this ribosomal RNA removal is targeting eukaryotic species, but by doing this dual depletion, we're also targeting the prokaryotic ones. Now, when you try to isolate circular RNA sequencing and you want to sequence that data, as I mentioned before, that circular structure they have is going to be very handy because we will be able to get rid of not only the ribosomal RNA, but also the linear RNA. And that's how you make a library that is strand-specific and only contains information the circular RNA strands. Now, if it's small RNA sequencing, you actually don't need any fancy kit to do the removal. You just need to do a site selection because the small RNAs, they range between 18 to 40 base pairs. So just by performing the site selection, it will be enough to sequence them. And even though we don't need to make any extra step to get a directional library, actually it is a directional sequencing strategy because it's going to sequence only one direction, which is single N50. And similar to non-coding RNA, you have exosome samples, we can also accept and work with them. Now, if by any chance or by any other reasons, you choose to make your own libraries, that's also totally fine, as we can also provide sequencing-only services. Now that we have finished with the introduction, we're going to jump right in to the non-coding RNA sequencing bioinformatics pipeline. So it can be a little bit overwhelming now with all these different boxes, but if you split them in different sections, it will be much easier to understand. So first of all, you will always try to start with the quality control of your raw data, and then move on with the mapping to the reference genome. Once you have the mapped reads, that's when you can perform the annotation and all the analysis, such as quantitative analysis to measure the expression of the transcripts, functional analysis, and also qualitative analysis. So let's try to dissect this pipeline step by step. We'll start with quality control. Now, you have the raw data that is generated by the sequencer and is usually stored in FASTQ format. This is usually the format that you can see when you open the FASTQ format, and it will have 4 different rows. So the first row is actually information about the sequence ID and also includes information about the library index. The second row is the one that is going to contain the read bases. As you can see, and sometimes there's an N, which stands for an unknown nucleotide that hasn't been detected. And a third row that works simply as a separator and in some cases can replicate the first row again. Now, the fourth row is going to provide you the quality scores in ASCII format, and it will later be changed to a numeric value. In any case, if you're having pair N reads, then please note that you're not only going to get one FASTQ per sample, you're going to get two files, 2 reads, read 1 and read 2 for each of the samples. And note that each file can contain millions of reads. Now that we know what is the raw data, how it looks like, that's when we move on to the data filtering step. So we're going to use this software called FastP, where we're going to introduce the raw reads and we're going to get clean reads. And since this is just a filtering, the format of the files won't change. You can get a view about the error rate, the base content, and also the proportion of raw reads that were also transformed into clean reads. Once you have these clean reads, you're going to work with them for the downstream applications. And that's when we are going to do the mapping part. So for the mapping part, we're going to use this software called HiSat2. And this is the one that we're going to use for non-coding RNA data. However, I know that there's plenty of literature and comparison between different assemblers, different aligners, and you can actually check this review here. And according to your project, to your own experiment, you don't need to follow us exactly and choose this software. You can choose the one that fits your needs the most and the best. In any case, we are going to use HiSat2 on the clean reads, and it is a graph-based approach that is going to index the reference genome, and combined with the bow tie algorithm, it will provide really accurate results and in a fast and sensitive time frame. Now, the input that you're going to have here is the clean reads in FASTQ format and the reference genome, which has to be in FASTA format. The output that you will get is a BIOM files, which is a binary version of a SAM file, a sequence alignment map. Now, these BIOM files will be used for following analysis, but it can also be visualized using the integrative genomics viewer, where you can compare them against the reference genome or also against the different samples you have, just to see the differences between them. Now, in Novogene, we also provide a table that will showcase the mapping rate among the different samples and some additional information like the total reads, total map reads that have been mapped on read 1, on read 2, and also the percentage of mapping rate on the send strand and on the anti-send strand. Okay, so we're done with the mapping. We have our BAM files, and now we move on with the annotation. So in the annotation, first of all, as we are studying non-coding RNA, we want to identify them. And the software that we're going to use is known as StringTie. Once again, if you have a preference for another software, that's totally fine. So StringTie, it uses a network flow algorithm, which also includes a de novo assembly option, which is going to help us assemble the transcripts into known and also novel gene models based on the known gene annotations. In this case, we're going to introduce all sorted BAM files and also the reference annotation file, which can be either in GTF or GBFF3 format. And we will get a GTF file, which is obtained through the transcript annotation of the assembled and the aligned reads. Now, once we have the assembled transcripts, we need to merge them using a command in StringTie to get rid of the duplicates and redundant transcripts. And now, once we have the merged transcripts, that's when we can move on and do some further steps to identify and predict our non-coding RNA types. We're going to apply some filters, and this is usually the step that we do in Novogene. And the first step is going to filter out the transcripts with low expression level and single exon transcripts. Now, the second step, given the length of the non-coding RNAs, is to filter those transcripts that have a length exceeding 200 nucleotides. And the third step is to filter out transcripts that overlap with annotated exon region in database. In other words, if we find any transcript that is present and can be detected in an exon region, potentially these transcripts belong to a messenger RNA, so it is not a non-coding RNA, and we don't need it for our current analysis. So we're going to keep them apart, keep them aside, and not include it in this step number 4. In step number 4, we're going to keep the remaining transcripts and determine the coding potential, which is going to help us to define if the transcript is a non-coding RNA or not. So this coding potential filter is a very crucial step, and we're going to use different tools in order to determine if it's really non-coding or not. We're going to make a cross comparison with multiple tools, and we're going to have the ncI, the CPC, and the PFAM analysis. So ncI stands for coding non-coding index, and it's going to measure the adjoining nucleotide triplets and see if they actually code for a protein gene or not. In case they code for a protein coding gene, then they're going to be removed during this analysis. Now, the CPC stands for Coding Potential Calculator, and it's going to extend and assess the quality of the open reading frames in the transcripts, and again, compare them to certain databases to see if they're going to be translated into proteins. In this case, the database we use is the UniProt knowledge base. And again, if they match with any kind of protein, then they will be filtered out. Similar thing happens in PFAM analysis, where we're just going to see if the translated transcript actually belongs to a protein family or not. Now, once we have the results, the ones that have remained during the analysis, we can represent them through different figures. So here you have a Venn diagram, which showcases the predicted non-coding RNAs from different software, those that are unique from each tool, those that are shared between 2 tools, and those that are common in all 3 databases. Aside from that, we can also represent the different transcripts depending on the coding potential. So you can have messenger RNAs, those that have been detected and filtered out, and the non-coding RNAs, those that remain for the non-coding RNA detection. Also, there is a group here called TUCP, which stands for transcripts of uncertain coding potential, and usually we will classify them as regulatory function non-coding RNAs. Yeah. We have seen the non-coding RNAs, but as I mentioned before, in raw data, we also have messenger RNA information. So we can use those assembled transcripts that were previously provided by StringTie and compare them to reference transcripts with this software called GFF Compare. And it will help us to know if these novel genes that are present in our data are novel or annotated. So Novogene will provide you a table that includes information about the position of these novel genes and also the potential function that they could have. Okay, so once the annotation is done, then we can move on with the quantitative analysis, which is one of the most popular pipelines here. So quantitative analysis, you want to measure the expression of your transcripts, and the simplest way usually is to quantify non-encoding and messenger RNA by counting the number of reads that map to each transcript. However, you need to take into consideration these two factors. When you're trying to estimate the expression level, this depends on the read counts and the total reads sequenced. And then also these read counts depend at the same time on the total gene and the transcript length. This means that you cannot just take the reads of each transcript and directly run the quantitative analysis. It is essential to perform a normalization step. So that's how you're going to make the data comparable between and within samples. There are many normalization steps. You can choose the approach that fits the most for your data. And here are just a couple of examples. So if you're working with single end data, you can choose the reads per kilobase of exon per million of map reads normalization approach. And if you're working with pair end data, which is for our case, you can choose the FPKM approach. Now, the FPKM approach and the RPKM approach, they're going to prioritize first the sequencing depth normalization and then perform the gene length normalization. However, some literature showcase that these results don't tend to be very accurate, and you can choose a different approach, which is known as transcripts per kilobase million, TPM, which is a normalization step that works for single and pair end reads. And it's basically the similar formula, but it switches the priority. So it first normalizes the gene length, and then it normalizes for the sequencing depth. Now, just a brief reminder of what we have been going through until now. We got the RNA sequencing reads, we aligned them, and then we assembled them with string tie. We removed the redundant transcripts, and we got the merged transcripts. So these merged transcripts, again, are going through the string tie software, and we're going to perform the E option, which is going to run the expression estimation mode, and it's going to provide us read coverage tables, which is something that looks similar like this. Now, in Novogene by default, we're going to provide you information about the FPKMs, but if you prefer to choose TPMs, just let us know and we can switch the normalization step. It's totally okay. Now, once we have the FPKMs, they will be in a certain format that can be accepted by the Bolgon software, and this Bolgon software will provide you information about the differential expression. So in this case, this is some information that you can obtain from a string type, and this is just another information that you obtain after doing the differential expression analysis. So once you have a list of differential expressed genes, you can perform clustering and see how these transcripts group according to the similar expression. Okay, now by relying on the data that we got in the previous stage, the expression data and the differential expression data, we can perform some additional digging on it, and it's known as functional analysis. So what can we actually do? First thing, geo and KGG enrichment analysis. So we have a set of genes, we have a set of transcripts that are differentially expressed, but it will be really handy if we can assign the biological relevance on them. So it is a way where we can determine if these differential expressed genes, differential expressed transcripts have any known biological functions, are present in any sort of interactions or pathways. So what are we going to do? We're going to use the software cluster profile, which is going to implement a fancy method to visualize the genomic coordinates, the gene and the gene clusters, and it's going to enrich the data. So by default, we're going to use Geo and KGG, which are usually the most frequently used databases. They're very popular these days thanks to their long-standing duration and their ability to offer a wide range of species. But if, for instance, you're interested in other databases such as the DSGNet or the Reactome, just feel free to let us know and we can incorporate them during the analysis stage. Now, aside from the enrichment annotation, we can also do non-encoding RNA target prediction. So what this means as target prediction. As I mentioned before, non-encoding RNAs, they can regulate gene expression at multiple levels. So they can interact with proteins, DNAs, and also RNAs. And some of them, they actually act as cis regulatory modules, which means that they can activate or suppress the transcription of neighboring genes. So we have this figure here and the following definition. We're going to define a cis-regulated gene as a protein-coding gene that has been co-expressed with a long-coding RNA that is located nearby, which is usually 30 kilobases upstream or downstream and in the same allele. So it is quite interesting to study the long-coding RNA and messenger RNA co-expression to see if this messenger RNA can be actually a target or not. And this is something done by the Pearson correlation. And you can see here a table of results showing you the long-coding RNA and its corresponding messenger RNA. Okay. Now, aside from the table and the co-expression estimation, you can also visualize the interaction between long-coding RNA and messenger RNA through a beautiful network with a software called Cytoscape. So it is quite easy to do. First of all, you're going to, of course, find the protein-protein interaction information of your target species. And how can you find that? Well, in our case, we're going to just enter our query on the string database. Now, once the string database provides us the information regarding our species, we're going to extract it and import it to Cytoscape for visualization purposes. And then you can edit the settings and display the figure as you like. So in this case, what we have, it's a long-coding RNA and messenger RNA interaction network, and the different nodes corresponds to the long-coding RNA and messenger RNA, and the lines indicate the interaction between them. Okay, so we're done with the functional analysis, the quantitative part. Let's rewind a bit and remember that in the mapping to reference genome stage, we generated some BAM files, and these BAM files can be recycled to do some qualitative analysis. So let's see what we can do. First, we can do variant discovering, also known as variant calling and annotation, with the solver GITK. So it's going to identify where the align rates differ from the reference genome, and it's going to generate a beautiful table known as BCF. You're going to introduce those BAM files, and you will get the BCF files. Once you have the BCF files, you can use different solvers. In our case, we use SNP-F to perform the functional annotation of these variants, and it will provide you a table like this, telling you the position in the chromosome and in the genome where this SNP occurred, also the quality of the variant calling, and some additional information regarding the gene, the feature, and the biotype. Now, aside from the variant discovering, you can also perform alternative splicing using the solver RMATs. RMATs is designed for detecting differential alternative splicing events in replicate RNA sequencing data, and it's going to do the classification, quantification, and expression level analysis of different alternative splicing types. Now, I'm not going to be very detailed in this part because we previously mentioned in our RNA sequencing data results webinar. And if you're interested, please feel free to check that out. Okay, so we are done with the detailed view of the long-coding part. What about small RNA and circRNA? We're going to take a quick view about them. So it's going to follow a very similar structure as before. So please don't be overwhelmed by this workflow. Again, we're going to have a data quality filtering process, moving on with the mapping as we get the BAM files, and do some basic annotation. Once the annotation is done, you can choose quantitative analysis approaches, quantify the small RNA, and then do some functional analysis. And that's it. So it's not as complex as it seems. It's just that it might involve different softwares and you get different results. But the overall structure is the same. Okay, so small RNA sequencing data, what it looks like to do a bioinformatics service with Novogene is that we'll provide you some basic information like the length filter. So we will tell you usually in the distribution of the small RNAs detected and give you an estimation about potential miRNAs, sRNAs, and pRNAs. But of course, this will depend on the annotation stage to verify these results. Now, in the mapping side, we're going to do the mapping with the Voltai software. And that's because it seems to be giving really good results and give us a zero mismatch alignment that other softwares fall short in some cases. Again, if you're working with a different software and it works well for you, that's totally fine. In any case, once we have the mapping done, that's when we can move on to the annotation and do some basic identification of miRNAs. For instance, if you want to see some annotated and well-known miRNAs, we can directly rely on the miR base But on the other hand, if you want to do some novel miRNA prediction and also estimate the secondary structure prediction, you can rely on the software miREvo. Now, another interesting feature that is exclusive of small RNA sequencing is the miRNA base edit. Because not sure if you know aware of this, but the small RNA, the microRNAs, they have a seed region. This seed region is usually taking the position of between 2 and 8 nucleotides, and it's highly conserved in eukaryotes. So this seed region usually actually determines the target of the microRNA. And if there's any changes on this seed region, it will probably change the target of the microRNA. So it is quite interesting to study to maybe identify potential biomarkers of diseases. And an easy way to do this is to align unannotated small RNA reads with mature miRNAs from the miRNA base. Now, aside from the base edits, you can also do some further exploration, like maybe study the overall miRNA family analysis, and also perform the usual quantitative and functional analysis. So if you're working with replicates, you can use dSeq to perform the differential expression analysis. But if you don't have any replicates, then we recommend HR. For target prediction, again, you're going to rely on a similar way as lncRNA with messenger RNAs to predict target genes and the relationship between miRNAs corresponding target genes. Okay, so we're done with small RNA sequencing, and we'll move on with circRNA data analysis. Once again, this is the full pipeline, and we can divide them in data quality control, mapping, annotation step, quantification, and functional analysis. It's all the same. So the mapping here is going to be done all with the software star, and we can provide some additional information like the distribution of circRNA's length. And when we are trying to do the identification part, we're going to use the software circExplorer2. So this is a very powerful software that is going to report the circRNAs that match exactly as the existing gene annotations. If by any chance you want to be a little bit more loose and not find the exact boundaries, then you can visit the circExplorer2 website, as they have a guideline for those cases. Now, this software helps us to characterize circRNA and also backsplice circRNA. And aside from that, you can run the general quantitative and functional analysis, again with dSeq2 for replicates or HR for non-replicates, and geo and KGG enrichment analysis. And usually the differential expressed genes can be represented by different figures, such as volcano plots or Venn diagrams. Okay, an interesting point that I would like to mention within the circRNA sequencing data analysis is that, you may remember this, one of the main functions of circRNAs is to act as microRNA sponges. And by doing this, they're going to inhibit the repression of gene expression. So it is very interesting to study the interaction and the relationship between microRNAs and circRNAs. Can we do this? Yes, we can actually annotate the pairs of circRNA and microRNA by using different softwares. If you're studying animals, then we recommend the software Miranda. And if you're studying plants, we recommend the PS Chromosome. You're going to get a table with the list of interaction of microRNA binding sites. And with this, I would like to move on to the last part of our webinar today, which is the non-coding RNA interaction. Because circRNA is not the only molecule, the only kind of non-coding RNA that is interacting with microRNAs. Actually, there is this theory known as the competing endogenous RNAs, which says that non-coding RNAs, messenger RNAs, and circRNAs have a competition relationship trying to interact with the microRNAs. And depending on who interacts with which type of microRNA, is going to change the gene transcription and the transcription regulation. So can we actually provide some information about this? Yes. So this is how we do this. This is a pipeline that can be applied when you perform whole transcriptome sequencing. This means that you have data of non-coding RNA, messenger RNA, small RNA, and circRNA. And it will provide you a comparison against all the different types. So here you have an interaction analysis of non-coding RNA with messenger RNA. Then on this part, you have a comparison between circRNA and messenger RNA. And last but not least, you have an interaction analysis between all the 3 different types. So with this, I would like to conclude the webinar. And once again, thank you all for your attention. and we'll just move on to the Q&A part to answer some questions from the audience. Thank you.
Thank you so much, Carolina. That was excellent. So yes, we have had some questions come through. So I'm going to start with this first one from Agnieszka. So she's asking, to analyze long non-coding RNA, do we need to perform separate sequencing, or can it be done together with coding RNA?
It can be done together. So you will probably see in the slides that the ones we share after the webinar is over, but long non-coding RNA is isolated through ribosomal RNA removal. So we're just getting rid of the ribosomal RNA and keeping the rest. So in the data, you will have messenger RNA and long non-coding together.
Great, thank you. So next is from Bered. What is the size selection value for sRNA sequencing?
So for small RNA, usually the size selection in the library preparation is 18 to 40 base pairs. But if for any reasons you want to modify that, you can let us know and we can accept that request.
So next we have a question from Stephanie asking, is it possible to identify long non-coding RNA without a reference genome?
Yes and no. So first of all, when you have long non-coding RNA, it is very important to have the reference genome when you want to do the long non-coding RNA prediction. But we can use a previous step of de novo step and trying to assemble a transcriptome. And then once the transcriptome is assembled, then we can use it for the downstream applications. So we might need to incorporate an additional step on it.
Great. Next question is from Laura asking, can we use the same pipeline analysis for mRNA-seq?
Yes. So the softwares that we choose in this case are targeting the long non-coding RNAs and are the most suitable for them, according to the literature. But you can also use this for messenger RNA, and perhaps in some cases, like instead of string tie, maybe you can choose a different assembler that works better. But overall, if you want to follow this, it's totally okay.
Lovely. Next question is from Larissa asking, what happens if I cannot provide a biological replicate?
It's fine. To be honest, we always recommend sending biological replicates, but if by any reasons you cannot do this, then we have alternative solvers that work really well with no biological replicates projects.
Great. Next question is from Raman. Is there any evidence that the small RNAs, such as long non-coding or circular or others, aid the function of mRNA, and filtering the smaller ones might alter the function of mRNA?
Sorry, could you please repeat that?
Sure. So they're asking, is there any evidence that small RNAs, such as long non-coding and circular, aid the function of mRNA, and that filtering the smaller ones might alter that the function of the mRNA?
Yes. So within small RNAs, we have this type called microRNA miRNAs that they can silence the messenger RNA expression. So there is evidence suggesting that there is this competitive regulation going on. Then when we have long non-coding RNAs, they can actually bind to the microRNA and avoid that gene silencing going on. And the same case happens for circular RNAs. It is actually known as the competing endogenous RNA theory, and there's a lot of publications regarding it.
Okay, great, thank you. Next question from Lydia. Why filter out low expression transcripts? Because aren't long non-coding RNAs usually expressed at low levels?
In this case, where the threshold that we are setting is really low, so it's for those cases where we consider the transcript to be not really informative. So maybe in those cases, it's not only low expression, but maybe there's only one single exon being expressed, and we are just setting the threshold to expressing more than one exon. So if it's just two exons being expressing, that's already being included during the downstream applications.
Okay, thank you. Next question from Athena. Is it okay to compare TPM values from different tissues and different runs? So for example, can we say that gene X with 100 TPM in the head is more expressed than the same gene with 20 TPMs in the liver?
That's a tricky question.
Yeah.
So if you want to compare them according to the tissue, you could say that it's been more, the TPM works, the normalization works, but given that they're in different tissues, the observation that you're making here might not be accurate. So the cells that are involved in the different tissues are different, hence the differential expressed genes that you find might be caused to different reasons. So in order to do that, instead of doing bulk RNA sequencing, it might be worthy to consider single cell RNA sequencing approaches, which is something that currently we cannot offer, but we can accept sequencing 10x or other single cell library types and perform also the bioinformatic analysis for you.
Okay, great, thank you. Next question from Vered. How do you handle annotation, quantification, and network analysis for non-model organisms, or for cases where the data is sparse?
So even if they are not non-model organisms, as long as we have a good reference genome and a well-annotated reference annotation file, we can perform the analysis. They don't need to be necessarily model organisms.
Okay, great. Next question is from Howard. Are there big differences of non-coding RNA between human and mouse samples?
I'm afraid that there could be situations where there are big differences between human and mouse non-coding RNAs, but it will really depend on the context of the project.
Okay. Next question is from Yue. Can we send FFPE material?
Yes. So actually the library preparation that we have, which removes the ribosomal RNA only, works pretty well for FFPE samples too, even if you are not interested in studying non-coding RNA. Like if you want to do messenger RNA, will also be the most suitable approach, because messenger RNA have the option to do poly-A enrichment library, but because FFPE samples are degraded, they are usually have, it is really hard to detect the poly-A tail. So regarding of the type of long RNA that you want to study, the non-coding RNA approach that we are mentioning in this webinar is the best one.
Okay, great, thank you. Next question from Samantha. How can one predict a function of a newly annotated gene?
So we are trying to, so if the novel gene hasn't been annotated, it means that we couldn't find any reference annotation there. What you can predict is relying on the differential expression analysis. So as I mentioned before, you can go and perform the clustering analysis of the differential expressed genes, and by seeing nearby genes that have a similar expression, you can assume that they may be involved in the same pathway. And in order to answer that question, you can perform the enrichment analysis, the GO and KGG, to confirm if the gene could be related in that biological process.
Great, thank you. So I think that's everything, all the questions that we have commenced. So I think it's time to wrap up. So I would like to just thank everybody for your time today. And thank you, Carolina, for such a great presentation. We hope you all enjoyed the webinar and found it useful. And thank you for all the questions too. They're very, very thought-provoking questions. As mentioned, if we haven't managed to answer your question live, we will get back to you by email next week with the answer. In the meantime, we will email you a link to the webinar recording and the slide deck, along with a short survey that we'd appreciate you taking the time to complete. The survey will help ensure that we make future webinars even more relevant to you and your research goals. There will be a link also in the follow-up email for you to view a certificate of attendance if you need one. In the meantime, please stay in touch. You can follow us on Twitter and LinkedIn, or you can get in touch via our website at novagene.com. So for now, goodbye and thank you, and we look forward to seeing you all on our next webinar.